probe design for in situ hybridization. In order to detect target clone, the construction of probe, it is very important. If we want to construct probe, then we must have the idea about the sequences of the target gene. If we have the necessary information, then we can construct the probe. As I told you that probe is a segment of DNA, or it may be oligonucleotide that may be variable in length, and it is complementary to the target gene, and length may be around 40 base pair, maybe 100 or 1000 base pairs, and finally it is used to detect the target gene. If we want to detect the complementary sequences in the target gene, then hybridization by using the probe, it can be performed at high stringency. That is, very stringent condition can be used, so that only the complementary sequences in the probe and the target gene may be hybridized with each other. High stringency means that we can increase the temperature for annealing, or melting temperature can be increased, and the ionic strength of the salt, it may be lowered, so that only the sequences that are complementary, highly complementary sequences, in other words, they can base pair with each other. On the other hand, if we want to detect just related sequences, then the hybridization, it can be performed at low stringency, like temperature can be lowered, so that those sequences that are closely related are in other words, some unrelated sequences, they can also form base pairing. So at low stringency, highly related sequences and some partially related sequences, they can also hybridize. So in this way, at low stringency, we can also detect homologous genes in a particular species. Suppose we have constructed a probe from a gene that is highly conserved, and it will base pair only with related sequences. But if low stringency conditions are given, then some other gene families that are homologous, they can also be detected. So this strategy of low stringency has been used to detect Hox gene in different type of organism. Hox gene are those genes that can control developmental processes in higher organism, like in human, like frog or other vertebrates, and even in frogs or insects, these Hox gene can be detected by using a particular probe. So cross hybridization may be there at low stringency that can give information about the other closely related gene families. So low stringency in this way can be used to detect closely related gene families. On the other hand, if we want to use, or if we want to detect closely related sequences, then the hybridization can be processed at high stringency. Sometimes these probes can be synthesized under the laboratory conditions, because sometimes if RNA probe is not available, DNA probe is not available, then it can be synthesized. And for the synthesis of probe in the laboratory, we can take help from the amino acid sequence. So amino acid sequence can be converted into a nucleotide sequence, and then it can be used for the construction of probe. But if we are going to construct DNA probe by using amino acid sequence, then we must take into account the degeneracy of the amino acids. Degeneracy is a situation where more than one codon can be used for a particular amino acid. So in this case, we have to use degenerate probes, like the degenerate primers. And here if certain amino acids are there like the methionine or tryptophan, then it is more valuable, because these two amino acids, methionine or tryptophan, they are coded mainly by one codon, so their incorporation into the degenerate probe, it may be more valuable. So degenerate primers can also be synthesized, and in addition to that, degenerate probes can also be constructed from a particular amino acid sequence. So in any case, if we are going to construct degenerate primers or DNA probes, then we must take into account different factors like the melting temperature, annealing temperature, overall length of the probe. So all these factors can be taken into account. So in order to perform in situ hybridization, we need to develop a particular probe, and this probe can be developed if we have an idea about the target gene, that what may be its sequence, or in other words, the amino acid sequence can also be used to construct degenerate probes.